Hello, everybody. Welcome to Bug Hunt. Y'all are too sweet. Thank you so much. Yes. Oh, Roxers, thanks for the resub. Hello, everyone. Hello. Nikki, Lucky Lou, Araxin, Russian, Sam, Dan, Perkachu, Null Zilch, Roach, Cosmic Cat, Siesta, Vic, hello everyone. Posh, thank you, everybody. Yeah, I'm old now. I turned 30 today. But couldn't think of a better way to spend it than searching for bugs in the pasture with you all. So, welcome in. Thank you for all of the birthday wishes. Yeah, let's get this started. Let's see what we got. Al? No. Virgos ended yesterday. I'm a Libra. Michael Russell, thank you. Still catching up. There's so many of you guys tonight. God, thank you, everyone. Welcome to the club. Old, yeah. Birthday buggies. All right. So, how's everyone doing? Anyone find any exciting bugs this week? Oh, our dung beetle boy's back. Ooh, there's a it went under. Sharp Easy. Hello. Dung beetle's going. There's a lot of action on the poo this week. The ants are back, which is not ideal. Hi, Mom. That one was quick. He jetted across. There was another cool beetle that popped up onto the screen there when we first started. There was a rove beetle, which is a very elongate thing. They have very small... That's him in the middle. If he comes up... Yeah, this guy. That guy right there. So that's a rove beetle, which is the family Staphylinidae. And you will often find them on poop, or like with poop. However, unlike the dung beetles, they're not there specifically for the dung. The whoa, shaken. Thank you, Tortilla, for the link. Yeah, so Staphylinids are actually the most diverse group of insects on the planet. There's something like 63,000 or 60,000 described species worldwide. They have these very small elytra. So most beetles have very long wing covers that will completely cover the abdomen, which is really helpful because it's protective, but it does limit mobility in some ways. So rove beetles have evolved very, very small elytra that sort of looks like they're wearing a backpack. And the reason that they have those small elytra is because they actually have a Staphylinid specific fold in their wings. So they're able to fold their wings up another good time, or another time to be able to tuck them up under those very small elytra. And poop was the first line. Nicole should be used to it by now. So they have these very small elytra that they're able to tuck their wings up under, but what that allows them to do is move into very small habitats, still have their wings protected, but they're very highly mobile. So they're able to live in a lot more places, and so because of that, they have diversified a ton. So, like I said, there's none that are specifically attracted to the dung itself. They're not doing anything with the dung like the dung beetles are. They are not using the dung directly for their young, but rove beetles are predatory. And so they will go to the dung for the flies and things like that that are actually in the dung. So rove beetles will oftentimes show up at dung because they are trying to feed on other insects that are there specifically to feed on the dung. Took off. We were getting a nice look at that guy, but then he's gone. But yes, so they're there eating the bugs that are eating the poop. So it's like a second or third level of attractant there to bring them in. So it's not uncommon to see them on the poop, but there are a ton of different species. So we saw one there as it was going that was pretty large. And so we were able to actually pinpoint that one, and we got a little bit of a good view of it. But with 65,000 or whatever it is species worldwide, there are a ton of them that are very, very, very small. So I would bet so much money that there are rove beetles here almost every week, if not every week. But a lot of the time they will most likely be too small for us to actually catch on the cameras. These cameras are phenomenal. Obviously, we're able to see tons of stuff from very, very far away, but a lot of the time those rove beetles would be under the dung, so unless somebody's there to flip it over for us, we wouldn't be able to see them. But even if they were up above ground, you saw how fast that big guy was. So pretty hard to track down, but that was cool that we got to see that. I don't know that we've had the I don't know that we've actually had like an identifiable one on here before. Hawk raptor. Yes, incredibly difficult. You need a microscope for sure. You also probably need to dissect them. That's a big thing that I've talked about in terms of identifying things here, but also over on my streams, which I do have a microscope hooked up to the stream itself. We're able to see some of those very, very small characters, and there's a lot of male genitalia examination that goes on with identifying beetles. The reason for that is that, like, insect genitalia is often described as like a lock and key mechanism, where if you have your car key and your house key, your car key is not going to unlock your house, and your house key is not going to start your car. So when the genitalia is compatible, that means that they are the same species. And when the genitalia are different, that generally means that they're distinct species from one another. And so as invasive and time-consuming as it is to dissect some of these very, very small beetles, sometimes that is like the only way to identify them. And historically, a lot of folks who are doing species descriptions would only focus on the male reproductive parts. So if you had a female specimen of one of these species, you might not be able to identify it given currently available resources. DNA typing things and DNA sequencing them to match is becoming cheaper and cheaper, but it's not yet at the point where, like, you could send something in for fun for like $5 or $10. It's still relatively expensive, but much, much cheaper than it used to be. But still, as a person, until we're able to readily interpret that sort of information with like, you can read the DNA sequences, there doesn't need to happen, but it is still a lot easier to get expertise and differentiate species with some sort of physical character, whether that be external or whether that be via dissection. There is still a physical difference between a lot of these things, and there was a big controversy a few years ago where somebody went and described like 400 species of wasps in one paper, but they only described them using DNA sequences. There was no physical description at all. They were just like, yeah, this wasp has this DNA sequence, and this wasp has this DNA sequence. And that caused a real big rift in the science world of like, is that an acceptable practice or not? I generally lean the way of like, no, that's lazy. And again, because that's not interpretable by humans, it's kind of useless. You have all these big computer programs that are able to help you understand things, but there is still an output that needs to be interpretable by humans. And so just having a DNA sequence in terms of like having that be the defining character is bad. I just think that's just straight up bad. So that was a bit of drum a few years ago. There's been so many rebuttal papers written about that. It's a whole thing. And yes, I do believe that was a katydid while I was on my wasp rant. Nicole, some trees hybridize, you just gotta figure it out. Yeah, birds do that too. Hummingbirds in Arizona in particular, there's a ton of overlap because there are like 14 species of hummingbird that occur in Arizona. So you get lots of different combinations, and because there's such a dedicated birding community in Arizona, people find them a lot. And so you're like, oh man, yeah, it looks like this species, but it's got characteristics of this other one. It's like, oh well, they probably did hybridize. Hybrids, for anyone unsure, a hybrid is when two separate species produce offspring. Now, using something called the biological species concept, it would be the blue jay, green jay. Yeah, I did see that. That's basically, what we're getting into right now. Under the biological species concept, the lowest definition for what a species is if two individuals are able to reproduce with one another. But you can have pretty sufficient overlap and still be able to reproduce, but also sometimes those hybrids are not viable. So like lions and tigers can mate and they can produce offspring, but those offspring then can't mate again. So they're sterile, and that pretty as much as like, yes, these are separate species because the offspring of this group can't mate. But with plants, it gets super weird because oftentimes they will be able to mate or, breed, pollinate each other, whatever. They will cross, they will have characteristics of both parents, but they will have reduced fitness compared to the parents, meaning that like they wouldn't produce as many fruits with as many seeds. They're not as big. And so where do you land on that with the biological species concept of like, yes, it produced an offspring, but that offspring is really bad at being whatever individual that it is. And so defining a species is actually really difficult, because under the biological species concept, again, if that's something that can reproduce, well, what about all the lizards and all the praying mantises that can reproduce parthenogenetically, meaning that they're all female and they don't have any males to add populations? That's not breeding, that's essentially cloning. And so where does that fall under the biological species concept? There are other species concepts that people use, but biology is very much filled with gray areas. There are exceptions to almost every rule, especially in entomology, because insects are so diverse. There's like one freak somewhere that's doing something weird that nobody else is doing, and you're gonna find that in bugs most of the time. Yeah, exactly, Nicole. Cosmic Cat, we are all on the new species side of the internet. Yes. I'm sure we are. We're definitely like plugged into that side of things. And I will do a bit of a shameless plug for a friend of mine. There is a podcast out there called the New Species Podcast. And she interviews scientists who have published papers that were describing new species. So you get to listen to their background of like how and where they found it and their justifications for, calling it a new species rather than it being one of the existing ones. Going through the process, listening to some of their fieldwork stories and things like that. I did do an episode of that podcast a while back that was focused on insect collections. The friend who runs it is an entomologist, so it's obviously very insect heavy. But there was just a new species of porcupine, I think, described, that she interviewed the people who did it. So, highly recommend that podcast. Definitely, like I said, shameless plug for a friend of mine and also an episode that I've been on. But I know there are several chatters who listen to and like that podcast, so if you're on the new species side of the internet, that might be a good place to go check out. I'll definitely put a link for that in the recap for tonight. This may be a dumb question. It's not, Murray. Feel free to ask whatever you want. They're your favorite bug. That's very cool. So yes, cicadas are out now. There are a lot of different types of cicadas. So a cicada is a whole family of organism. And some of them, like the ones that were out in Ohio, had the really big, like, 17-year emergences. And those individuals of those species do only come out every 17 years. But most cicadas aren't on a 17-year cycle. There are cicadas that are on like an annual cycle, so there are cicadas in areas every year, but certain species will have different lengths of time between when they come out. There are 13s, there are 17s, there are like 6-year cicada, or there are 7-year cicadas. So it's very possible that they're out now. Not sure where your area is, but it is probably getting to be the end of cicada season because they do tend to be out more in the summer. W.N. Besser, can viable hybrids reproduce with for a second level hybrid? Sometimes they can, sometimes they can't. And that's like one of the questions, right? Is like, can it reproduce again? And if it can, like, well then is it its own species or not? I think this is another new spider chat. I don't know that we've had these before. Yeah, new species podcast. Thanks for the follows, guys. I'm sure Zoe is going to appreciate that. Roxers, yes. Great call. This is a lynx spider. This is a male lynx spider. So you can see the pedipalps, those big kind of boxing gloves that are under the head. Those are the male reproductive organs. So spiders have indirect sperm transfer. So they will put their sperm down on a web and then pull the sperm up into these pedipalps, or these little boxing gloves. Yes, Tortilla. I call them sex mittens. They have a hypodermic needle or like a turkey baster type apparatus in there that they're able to pick up the sperm with. And then they approach the female from the front, go underneath into her abdomen, and then deposit the sperm that way. So they don't have like a direct sperm thing. There has to be this intermediate term here. But that's often how you can identify male spiders, is that they will have these big old boxing gloves on their pedipalps. Or, sex mittens, whatever you want to call them. I think sex mittens are funnier. But they use those to reproduce. So that's what you're seeing, those kind of dark structures that are moving around towards the bottom of the screen right now. The other way that you can tell, there's a couple of things that are good for identifying lynx spiders. Something that we can't see right now is actually the eye arrangement. Lynx spiders tend to have a little bit of they have, the top of their carapace, but then their eyes sit further above that, on like a little bit of a dome. So that's something characteristic of this group. They also have very, very spiky legs. So it's kind of hard to see here with the IR, but when Logan was focusing it, I was able to see fairly large spines coming off of the legs, and again, that is quite characteristic of this group. So that's a neat little find. Good spot on that one, Logan. Good spot. So going back to the species thing, if we want to, and we can keep going on this if people are still having questions. Defining what a species is quite difficult, and most people don't think about that. Especially with insects, a lot of the time you will find new to science species in a museum. And that means that it is dead and has been on a pin for who knows how long. And with mammals and birds and all of this stuff, it's actually pretty easy to observe them in nature breeding or mating or doing whatever they're doing. With insects, it's a lot harder to track something that small, hard to figure out where and when and how they're actually mating. So that's why we have to refer to the actual physical structures of the reproductive bits, because we don't have the option to really watch them do it in nature. So we have to basically make that call and see if, yes, we think that these two, the structure of the genitalia might be different enough to warrant calling them separate species, or if they're similar enough to group them all as the same thing. But making those designations based off of specimens in a museum is kind of tricky. So there's definitely parts of it that come down to physical examination, but you can also then take pieces of that insect, usually a back leg, because that is usually where the largest thigh is, that you can get DNA work out of it. So DNA is definitely not the end-all be-all. It is a very, very helpful tool, but I personally think it is most helpful when used in conjugation with, in conjunction with physical characteristics and actually like looking at stuff. Museum comment made me think, do older museums have comically bad pinning like they're comically bad old taxidermy? Yeah. A lot of comically bad pinning that I've seen comes from students. People just learning things, you know. They'll put the pin in the wrong place, you know, because there are proper places to put the pins. You'll get stuff that's tilted really bad, stuff that's pinned upside down almost, like the pin is going through the side of it, and that's no good. Typically, we don't have those pulled out separately. Because the idea of the museum is that, like, if you're there to work on, you know, Group A, you should be able to go to the collection and find all of the Group A that we have all together. And so even though it might be one specimen that's pinned poorly and would make a funny display thing, unless we had more of that thing, we would probably keep them all together. Now, I personally have a small collection of things that students have turned into me as a joke, but I have one that is just a leg. It's just the leg of a cricket, and they pinned it, and the label says leg. I thought that was hysterical. But then someone also turned in like a little, maybe 2-inch piece of stick. You know, it's just like a piece of wood. They put a pin through it, and then they labeled it as Phasmatodea, which is the order for stick bugs, which is very funny. Like, objectively, very, very funny for them to have done that. Couldn't give them credit, obviously. But there have been some good ones. There have been some really good ones that students have turned in. And I thought the stick bug one was very funny. So I do have like a personal little thing of poorly done or just like funnily executed bits from collections that students have turned in. Cool moth that we had come in right there. Couldn't really tell what it was. But definitely going through student collections is not for the faint of heart in terms of wanting to see good pinning. No, Nicole, I don't think they were actually trying to get credit. I think they were being funny. Because someone in my entomology class, like when I was taking it way long ago, pinned a plastic cockroach in with all of their other specimens. Again, not trying to get credit, just kind of trying to be funny. And that happens more often than people would assume. People definitely try to pin fake bugs as a bit of a laugh. But grading insect collections is always a really interesting process. And when I was an undergrad, I TA'd for a class that had a collection requirement of like 100 specimens, but we also had this really full online class that these students were not on campus, so they never got the hands-on experience of pinning stuff. And they would then have to mail their collections to us at the end of the semester to get them graded. The worst thing that I ever saw was a piece of styrofoam inside of a pizza box, which, in and of itself, not the worst. We've had some good collections come in pizza boxes. However, this particular piece of styrofoam did not have insect pins. They didn't have any insect pins in the bugs in the foam. Instead, I'm going to give you just a second to prepare for what I'm about to say. We opened the lid of this pizza box. There is a piece of styrofoam. There are bugs on this styrofoam. Not thumbtacks. Not thumbtacks. Worse. Staples. I'm dying. I'm thinking about this. It's so funny. They opened up the stapler and stapled the bugs to the styrofoam inside of this pizza box. And that's just like the worst that I could not believe that in my 10 years of working with insects and working with student collections, that is definitely like the worst thing that I had seen. Man, I laugh every time I think about that. I tell students that story. I'm like, hey, don't be this guy. And I've never had anything that bad come in one of the classes that I was teaching. But yeah, that's still the craziest thing that I have ever seen in trying to grade student collections. That was quite wild. Marie, there's a Game Grumps insect pinning video. That's interesting. I am only like vaguely aware of Game Grumps content. And they don't seem like the types that would pin bugs. But I will definitely check that out. Okay, couple of questions on that. Why did that person bother? Truthfully, I don't know. I was not grading at the time. I was marking things whether or not they were identified correctly or not. So I don't know what they ended up getting. It could not have been anywhere close to passing. So I don't know really why they tried. I don't know what the plan was there, but it happened. They were just paper staples. I think what they did was they like opened up the stapler like you would if you were trying to staple a bulletin board, and just did it that way. So it wasn't even like the super long staples. What are the things that are graded on when it comes to collections? That's a good question, Nicole. Is the specimen prepared properly? So that kind of goes to like what we're talking about right now. They would definitely have gotten a ton of points off for specimen curation because their stuff was stapled and not pinned. But even when stuff is on pins, there are certain ways, like you're always supposed to pin off to the right-hand side. You're supposed to try and make things level and things like that. So we check that sort of stuff. We also check if it's an appropriate method of storing that particular insect. So things like termites dry out super fast if you try to pin or point them. So those have to get stored in vials of alcohol and submitted with your collection that way. So curation is a big one. Another one, obviously, is identifying the thing correctly. You know, can you tell me if this is a beetle versus like a wasp? And then even further, depending on the level of the class, you know, differentiating between a scarab beetle and a ground beetle and a ladybug, etc., etc. So identifying things is another big component of it. And then usually the last thing that we grade on is the labels. So if you've seen my streams where we've been looking at pin stuff, or any of the streams where we've talked about it before, there's a little piece of paper under every single insect specimen in a museum. And that little piece of paper tells you where and when and how and who collected that particular individual. And that information stays with the bug forever. So it tells you, like, oh yeah, collected this night, this day, in this location, etc. And so a long time ago, it would be fine to just say something like, oh yeah, this was collected in Arizona. Well, Arizona is a gigantic state, and nowadays we have GPS in our pocket, so you're able to get GPS points to basically send people to, like, I collected it under this individual rock. And you can send people back to that original rock if you wanted to, with very specific coordinates on your insect label. But that information is critical to have on there if that specimen is going to be useful for science, like, at all. There are lots of species around the world that look very similar, and regional identification keys will only get you so far if your specimen is in fact not from that location. There's a bunny. We got a bunny munching. So that label needs to stay with the specimen, and that's why it's on the specimen, like on the pin, for the rest of the life of that specimen. And without it, the specimens don't really hold much scientific value. It could still be a very nice specimen, you know, prepared well, really cool, big, flashy beetle, but without that information, it is not useful scientifically anymore. So we try to instill that when we're teaching the students, you know, labeling, making sure their label format is good and everything like that. But also, if they're saying that they collected a snake fly, for instance, which is a bug that only occurs on the West Coast, if they're saying that they collected that in Florida, well, that's a big no-no. We know that that would not be correct. And the same sort of thing if they were like, oh yeah, this beetle that only lives underwater came to, you know, I found it in the middle of the state where there's no water around. Falsifying that information is also very, very bad practice. So those are the things that we would tend to grade on when doing insect collections. The curation, the identification, and then the labels, which kind of gets rolled into curation as well. Moosums, pinning stream, maybe. I don't know if Maya would want to do another one on here, but over on my stream, which I've just started pretty recently, we definitely do some pinning and specimen prep, but the VODs are definitely there from Maya and Allison's pinning stream a while back. Parakeet. Read news about some Australian guy who stole butterfly specimens in the 1940s and painted on them and sent them back to museums so they were misidentified on display. That's very interesting. I'm going to have to look that up. Oh, okay, Nicole, good question. Would you say that some beetles or insects suffer from the charismatic megafauna thing, where if they're bigger, they garner way more interest, like the spotted lanternfly killing campaign? I feel like it was focused more on because of what it looked like. Yes, in some ways. It doesn't necessarily mean that it's bigger, but there are certainly insects that are more charismatic and people care about them more often. The biggest culprit for this is honeybees. In North America, honeybees are non-native, so they were brought over here by the Europeans in like the 1600s. And everybody is on this like save the bees kick, but honeybees are domesticated insects. So it's honeybees, silk moths, and cochineal scale insects that are the three domesticated insects that we have. And so Save the Bees, the original message was talking about native bees in North America, and everybody co-opts it into talking about saving the honeybees, which is quite frustrating because, again, they're non-native and they're a managed species. So my analogy for this is always like, oh yeah, we are, everyone's always for like, yeah, like let's save the bald eagles, like let's do that, let's save wild birds. The way to do that is not by raising chickens. And that's essentially what the program of like saving the bees and then focusing on honeybees is. The honeybees being the chickens in that instance. So I think bees are very charismatic. People know about them. They know about their behavior. They know a little bit more about their biology than most other insects. But I think they're a very bad ambassador species because they are non-native in general. Cochineal, the red pigment. Yes, exactly. Let me... Parakeet, Colin Wyatt. I'll have to look that one up. I didn't hear about this. This is a katydid. Yes, Bert, you can absolutely use that. Flum, good question. This is a katydid. So you see those super duper long antennae in the front? That's going to be a katydid trait. There are other groups. Crickets will also have fairly long antennae, but grasshoppers, your typical grasshopper, in the family Acrididae, 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 those ones are going to have fairly short antennae. Super long ones, almost always going to be a katydid. Additionally, everyone see that like kind of swooped up hook at the tip of the abdomen there? Little thing sort of looks like a bean or a banana, kind of going up to the right. What is it, chat? You know it. You know what that is. There it is. Okay, I was waiting. Not a peen this time, Bird. That's the straw ussy. For the uninitiated, the straw ussy is what we in this chat call the ovipositor. Ova meaning egg, and positor meaning positioner. So female insects will have a physical structure that allows them to place where the egg gets laid. So we know that this is a female katydid because we can see that structure coming out of the back. Now, that's another reason that we're able to tell that this is a katydid and not a grasshopper too, though, because katydids will have this very long, very obvious, like what we're looking at right now, ovipositor. while grasshoppers they're in a different suborder. So they actually have a fairly internalized ovipositor that gets averted. They are able to push it out and use it, but it is not always on display. They're not always on display with like this one is here on the katydid. Did Winnie make a noise? Or is somebody making noise? I don't know. I've got stream muted, so I can't hear what's going on during chat chats. I trust y'all. But yeah, so this is a pretty nice view of that katydid there. The big ovipositor also gives it away. But yeah, those super long antennae are a fairly good identification character for katydids. And if we were able to see the head a little bit better, we're not going to get it from this angle unless it miraculously turns around for us. But the antennae are also placed very high on the head and close together. So the antennae are usually going to be placed above the eyes and basically touching at the bases. In grasshoppers, the antennae are a little bit further down, a little bit further to the sides, and very, very widely separated on the head. So, again, lots of characters and things like that. Identifying things on stream here and with the cameras in general is kind of difficult because a lot of the things that are definitive characters about these groups are things that you need to look super close at or look at with a microscope. And so after having seen tons and tons of these things over the last 10 years, you start to get like a vibe for it, and identifications through pictures or even on these live cams a lot of the time is just purely like, oh yeah, like it's a katydid because I know it is, not necessarily because I can see all of the characters that I would need to identify it like officially. Yes, I know it's a katydid because of the way it is. It's just like, yeah, it looks like one. It looks like one, and I've seen enough of them to identify it as that. But there are actual physical characteristics that you're able to see, but not necessarily all the time with these cameras. I think we're pretty zoomed in, Russian. I don't know exactly how far we are, but I think we are pretty zoomed in. They get big, but they don't get that big. Okay. Logan's going to zoom out for us. So keep an eye. That's where we were. We were on that plant. So decently close to the camera and up, so we're able to actually get a pretty good view of it. So yeah, these cameras are crazy. Always do a phenomenal job of zooming in on all this stuff, and it's pretty incredible the type of stuff that we're able to see. I know several. Oh god, it was like way back. I remembered I was able to see a wing vein character on a fly at the on like the main pasture cam, and blew my mind. Lucky Lou, thanks for the raid. Welcome into Bug Hunts. We're going to be on here looking at bugs with the cameras for about another 10 minutes, and then we'll be back to the regularly scheduled Alveus programming. But please take a look at this bunny first. Munch, 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 munch. The fleas on the rabbit, yeah. That would be remarkable. I think we did on one of the rats. On one of the rats, I think we were able to actually see a parasite at one point or another. I think it would be much harder on a rabbit. Not, I mean, obviously it's kind of far away, but rabbits also have quite dense fur, and so it would be a lot harder to actually see parasites on a rabbit anyway. But yeah, I remember we saw a flea or a louse or something. I wasn't able to see it quite that well, but we saw bugs on one of the rats at one point, which was pretty neat. Again, parasites are not everyone's favorite thing. I just saw Marie say no. But I think parasites are super fascinating, actually. They're really, really neat with how well they specialize on certain groups. I think I've talked about it before, but one of my favorite collecting experiences that I've ever had was collecting Platycilus castoris, which is known as the beaver parasite beetle. It's a beetle that is super duper flat, it is eyeless, it is wingless, and was originally described as a louse. And the only place that you can get it, the only place that you can find this beetle is on live beavers. Or at least freshly acquired beavers. We won't go too into that story, but I have in fact collected them, and they're super sick. It's such a weird looking thing. And again, just like crazy how specialized those things are. Who studies eyelash mites? Is that entomology? Depending. Sometimes entomology departments have people that work on mites. That is technically acarology. Acarology. That is specifically the study of mites and ticks, but not everywhere has an acarologist like on roster, so that does fall into entomology sometimes, in the same way that usually entomologists end up having to field the spider questions as well. It's exoskeleton group, it's close enough for most people, so we kind of have a loose working knowledge about them, but not a super deep specific thing about them. Yeah, Bester, that's a great point. So interesting how they need to balance keeping the host functional and not getting too greedy, and also how the parasites have parasites. That's the whole difference between a parasite and a parasitoid. So a parasite lives on the host, and the idea is to keep the host alive as long as possible, because that's where you're getting your food. That's going to be your external things like lice and ticks and fleas and all those sorts of things that are feeding externally on these groups. But when you have wasps that are laying their larvae in caterpillars, or flies laying their larvae on ants or on leaf-footed bugs, those sorts of things are parasitoids, because they're actively killing the host. The goal is to feed the larva of whatever group you are, and it doesn't matter if you kill the host, because you only need that food source in your larval stage. So oftentimes you'll hear about people saying like, oh yeah, it's like that's a parasitic wasp, and it's like, well, it's actually a parasitoid wasp technically. because those are functionally very different roles in the environment. You know, parasitoids will always kill the host, while parasites want the host to stay alive so they can continue feeding on them. World's Wildest a bit ago where Maya said pubic lice are endangered. Is that still true? That's a tough one. Ooh, this is pretty. This is a really cool lichen moth. Oh, what's the genus? I know what this is. Moth. I should be able to find this one. This is very likely in the family Erebidae, which are the tiger moths. And there's a lot of them in this group that will have this very mottled Erebidae. And a lot of them will have this very mottled patterning, and it's because they are going to be resting on trees during the day, where they are going to be doing their best to blend in. So if you have this very mottled pattern like this, you're going to be blending into the chunky bits and pieces of lichen on tree bark and things like that, and moss. Casual, yeah, I think it is drinking the dew. So this is another case where entomology always has exceptions. Moths and butterflies as adults have a proboscis, which is a coiled straw-like mouth part. However, there are some very, very basal, meaning like early, like these are kind of the prototypes for what moths turn in, or what moths came from, that do have mandibles as adults and do feed on other insects as adults. However, this is not one of those. It's definitely not feeding ants or aphids. This is definitely just kind of sucking up those little tiny water droplets that are on these flowers here. It might be drinking honeydew from the ants, which I know is kind of a funny thing to say, since honeydew, people think about the melon a lot of the time. But honeydew is actually the sugary liquid secretions that aphids make. And that's the reason that aphids oftentimes get tended by ants, because the ants are there to take care of and feed on that very sugary liquid. But if there aren't any ants around tending it or whatever, aphids are very interesting in that they don't have the insect kidney system. So they don't really have any way to process all that extra liquid, because they're just feeding on liquid 24/7, or not 24/7, but like most of the day. And so they're able to kind of waste some of that sugar product by just drinking it and then excreting it through their little cornicles. They have these little tubes coming out of the top of the abdomen that are called cornicles, or my preferred term for them, siphuncuли. But that is where they are excreting all of this extra liquid that they're taking in by having a like 100% liquid diet. And because they are just drinking so much of it, they don't need to extract every last little bit. And so that's why the excretion that they have is quite sugary. And so it's possible that there were aphids on that plant above the moth, and then the moth was drinking some of that off of the plant, but I imagine that was mostly dew. Silvera, that is probably exactly what's happening. The oleander aphids on your milkweed getting tended by the ants. Yeah, I would bet that's exactly what the process is. She's so funky. That's a good one, Renshaw. I like that a lot, actually. All right, Logan, you want to take us back home, back to the pasture, see if we can get one more good spot in over there, and we'll wrap it up for the night. Went fast. Time flies when we're having fun, ain't that right, chat? Ooh. A mysterious flying orb. You're good, Logan. Take your time. We'll just look at this mysterious floating orb for the time being. The ghost. We're seeing some weird light reflection coming off of that, so I imagine that is something... Yeah, casual, exactly. I think that's something that is attached to a bit of spiderweb, and it's just kind of blowing in the wind. You can sort of see off to the left when it hits a certain part of the light. You can see little bits and pieces of web kind of going on there. Yeah, look at that. Look at that drag line. So she essentially, that spider attached the web line to the top of the building, and then I think probably leant down to try to go to the bottom. I don't know if that's like a glob of web glue or if that's actually the spider, because that web piece is attached further down somewhere. I don't know if we'll be able to track and follow it totally, but yeah, that's exactly what that was. That was, you can see it going up there too. Neat. I don't know if a Winnie Booger would get attached to the ceiling like that, Tortillo. But yeah, very interesting. Their drag lines can be super, super long too. The cool thing about the spider webs is that they can let it out, but they can also retract it in some they like are able to take it back up. They will actually eat their web to recycle it to be able to use it again. So a lot of the times to span these really big gaps, if you have like tree lines or whatever that you're trying to get to, they will drop to the ground, walk across, and then just climb up, and then basically take in all of the extra silk that they didn't need. Which is a pretty cool process, and we are always good to end it there on our dung beetle friends. Alright, y'all. We are going to call it there for the evening. Thank you all for being here. Thank you all for the very kind birthday wishes at the beginning of stream. As always, this is very fun, and like I said at the beginning, wouldn't have wanted to spend the evening of my birthday doing anything else. Always happy to do chats. Love talking to you guys, so this has been great. Logan, our wonderful cam operator. Please thank him as well. He is here every week, just as we are, but actually doing work while I am just talking about things here in the background. Join the bug cord, ! Bug cord. We have a Discord for the Alveus insect community. Something new that is happening in the bug cord is there is now a role selection. I've started streaming recently on my own channel and realized that there would be a lot of overlap between a personal Discord and the Alveus bug Discord. So instead of creating something totally new, you can go into the role selection and choose whether or not you would like to get my personal stream notifications. You don't have to. If you don't click it, you won't get the notifications and we won't bother you anymore if you would like to stay how it currently is. But if you enjoy the bug content, I do stream pre-chat chats. So for about 2 hours before we start here on this stream, I will do streams over on my channel, and I also tend to do streams on Monday nights, but I don't have a very strong set schedule yet. So check that out if you are interested. That bug cord is also good for the Chat Chats library, so I will post links and talk about things that we talked about tonight. The Alveus Clips channel, run by one of our community members. Excellent content on there, especially if you've missed recent collabs or fun animal scenes. Check out the Clips channel. And then, last but not least, stick around for rounds. We do check through all the animal enclosures before we all head off to bed. And one more look at the animals. Logan will need some help with that. So I shall call it a night here, and I will talk to you all next week. Thank you all again. Stay safe. Have a nice night, and I'll see you soon. Bye, y'all.